recurring journal club. My name is Alice Damatakis, and I'm the Senior Lead Scientist for Application Science here at Inscopix. We began this series at the beginning of April to help keep the scientific community connected during these difficult times. We'll be periodically discussing publications of importance to the field of circuit neuroscience, often featuring Inscopix and Vista or Invoke technology, and we're excited to have you join the discussion from your virtual offices at home. You can send us questions via the chat window throughout the presentation and for up to 5 minutes after the presentation is over. We will do our best to get to most or all of the questions with a hard stop right at 12 o'clock Pacific time. Today, our field scientific consultant, Jonathan Zapata, will be presenting a cell reports publication from Peter Kalivas' lab at MUSC. Jonathan joined the Marie Curie International Research and Training Program, SIMBAD, funded by the EU. He received his PhD in neuroscience from the University of Milan, Italy, and conducted his postdoctoral training in Dr. Nathalie Roche's lab at the Collège de France in Paris, combining in vivo imaging and in vivo electrophysiology in freely behaving mice. Jonathan is now working as a field scientific consultant in South EU and Scandinavia at Inscopix. And with that, I'll go ahead and hand it over to Jonah.
Thanks for the introduction, Alice. And good morning or good afternoon, everyone. So today I will present this paper entitled Opposing Regulation of Cocaine Seeking by Glutamate and GABA Neurons in the Ventral Pallidum from the Kalivas lab. So published in Cell Reports. I have decided to present this paper about cocaine addiction because addiction is still a major worldwide health problem. And to give you recent numbers, 35 million people worldwide suffer from drug use disorders, while only 1 in 7 people receive treatment. About cocaine, cocaine is used by an estimated 18.2 million people worldwide, and only about 0.4% of the global population aged between 15 and 64 years old. It is then important to better understand and dissect the addiction circuitry. In addition, this study used innovative and recent tools to identify and manipulate neuronal population that I would like to share with you. I would like to start with a brief introduction and describe the neuronal composition of the ventral pallidum and its role in addiction in rodents. The ventral pallidum plays a central role within the ventral basal ganglia, where ventral pallidal neurons' activity are necessary for motivated drug thinking in animal models of addiction for all known drugs of abuse. The VP contains 2 distinct neuronal populations that constitutes 95% of all VP neurons. Glutamatergic neurons that we will identify as VP-GLU in this study, and GABAergic neurons that we will identify as VP-GABA in this study. Activation of VP-GLU neurons is important to drive aversion, whereas VP-GABA neurons stimulation motivates reward thinking. These effects are mediated in part by projection to the ventral tegmental area, lateral habenula, and thalamic nuclei. Previous studies from the Kalivas lab have indicated that a subset of GABAergic neurons express the neuropeptide enkephalin, that we will call VP-PENK here for proenkephalin, where enkephalin signaling is necessary for the reinstatement of cocaine thinking in animal models of addiction. However, the role of VP-GLU, VP-GABA, and VP-PENK neurons in cue-induced drug thinking remains unexplored. The VP is the primary output of the nucleus accumbens, which in turn plays a central role in addiction. This pathway is composed of dopamine D1 medium spiny neurons, receptors, D1 and D2 receptors expressing GABAergic medium spiny neurons called D1 and D2 MSN in this study. And D1 and D2 projection show opposing information to the VP regarding drug thinking, D1 being involved in cocaine behavioral sensitization and cocaine syncing, whereas D2 MSN are involved in cocaine withdrawal-induced anhedonia and cocaine syncing extinction. However, the relative innovation by D2 versus D1 MSNs onto the different VP subpopulations is unknown. Therefore, in the present study, Jasper and collaborators have decided to dissect the subcircuitry connectivity and functional roles of the VP-PANK, VP-GLU, and VP-GABA subtypes during a cocaine syncing paradigm. Authors started the study by using in situ hybridization of mRNA encoding VPANK, VUGAT, and VUGLUT to quantify the proportion of each subtype in the dorsal VP. So in panel A is a representative micrograph of the triple in situ hybridization, and panel B shows the relative density quantification. They found that the dorsal VP is composed of a major population of GABAergic neurons, around 75%, and glutamatergic neurons, around 25%. When looking at the VUGAT positive population, around 20% of VP-GABA express p-enkephalin. As the dorsal VP received inputs from the core of the nucleus accumbens, they then decided to use a retrograde rabies monosynaptic labeling strategy to identify the relative D1 versus D2 MSN innervation. For this, DRD2 GFP mice were crossed with mice expressing Cre recombinase selectively in each of the three cell types using IRES Cre lines. To do so, they first transduce VP cells with rabies helper virus constructs, the receptor, the TVA, and the glycoprotein here, RG, and then transduced VP cells with a pseudotype replication deficient rabies mCherry. This is a very good tool that limits rabies virus infection to a small population of Cre-expressing cells and their directly presynaptic partners. Panel D showed a representative micrograph showing rabies-infected D1 MSN in red, so the D1 positive rabies positive, D2 MSN in yellow, that you can see here, and the proportion of D2 cells that do not innervate the dorsal VP is basically in green. When looking at the proportion of D2 cells labeled in green versus labeled in red versus labeled in yellow, what they found is that VP-GABA were innervated equally by D1 and D2 MSN, so you see that the proportion is equal. However, VP-GLU and VP-PANK, so around 84% for VP-PANK, receive more D1 MSN afferences. So in conclusion, the dorsal VP contains a large proportion of GABA neurons, and VP-GLU and VP-PANK receive major afferences from D1 MSNs of the nucleus accumbens core. Interestingly, previous work from the Kalaevas lab have shown that activation of the D1 MSN pathway in the nucleus accumbens core and their projections to the dorsal VP are necessary for cocaine-associated cues to initiate drug syncing after extension learning or training. Therefore, authors decided to run a similar training and combine a chemogenetic strategy to activate the different VP neuronal subtypes in Cre mice. To do so, they use VUGLUT, VUGAT, and VUGAT, VUGLUT, and PANK IRES Cre line. And in the first step, mice were injected with a fluxed or non-fluxed GQ dread into the VP. So here you can see a staining of DAPI, so to label a nucleus. Substance P was used to locate the VP. And in GQ dread, so here they used, it's not really GQ dread here, it's, sorry, it's in red. Then mice were allowed to self-administer cocaine for 12 days with an association of light-on Q pairing to each cocaine infusion, followed by an extension an extension training around 10 days without cue presentation or infusions of cocaine. GQ-DREAD was activated using a systemic injection of clozapine N-oxide, CNO, 1 milligram per kilo, prior to the late extension session or before a cue induced cocaine sinking. First, to confirm GQ-DREAD activation of VP, they injected mice with CNO or vehicle for control and immunostain for expression of the immediate early gene product FOS 2 hours after. So here they basically use the GQ-DREAD MC-trin for the total VP, so the whole VP, and for the different subpopulation they use GQ-DREAD M-sharing. So here, when you look at the quantification, you see that you have a significant increase of FOS following CNO injection, no difference in expression of MC-trin, and during the double quantification, you see that there is still this significant increase. When you look at the different here subpopulation of the VP, you can see that you have a significant increase of FOS expression in VP GABA, VP PENK, and VP GLU. When you look at the expression of GQ-DREAD, you don't have any significant difference between the expression of GQ-DREAD. That's good. When you look at panel G here, FOS plus DREAD co-expression, you see that CNO significantly induce FOS in various infected neurons across all cell type. Here, panel H represent non-infected neurons where FOS activated is significantly higher numbers following stimulation of VP GLU, suggesting that VP GLU neurons may directly innervate other VP neurons. So that's only in the non-infected neurons and basically the effect of the activation of other neurons. And finally, in panel I here, we can see that also it's FOS plus DREAD, that a large population of VP PENK neurons here were FOS activated by CNO, indicating that this population may be more excitable or responsive to the GQ pathway, signaling that VP GLU or VP GABA. So in overall, all VP neurons transduced with the GQ-DREAD show significant increase in FOS, and the use of GQ-DREAD is a good tool to activate neurons in the VP and VP subpopulation. So in the first step of experiment, so wild-type mice were injected with AAV2-HSyn and GQ-DREAD M-citrine. So here it's pseudo-colored in red for clarity. In the VP, so regardless of the subtype, so here, like the previous experiment, it's the whole VP was infected. We don't want to look at the different subpopulation here. When looking at the number of response, mice acquire a stable cocaine self-administration. So you see that you have active response. So here by active response, the active response is basically when the mouse no smokes. And you see that you have an active response, so with infusion of cocaine. When you stop infusion of cocaine, you have this increase in response in the absence of cocaine reward or a drug conditioning use. So here they look at two different time and two different kind of context. So one during extension and one during the reinstatement. So reinstatement, it means that the mouse has to no smoke and to basically receive the cues. So what we observe here is that stimulating VP neurons increases active no spokes during extension as well as during reinstatement, so during the cue-induced cocaine sinking. And during and the number here are that's also present in this histogram. So you can see the number of total responses, but also the number of active and passive. So passive, why? Because the mouse has to basically choose the right let's say no spoke to receive the reward. So inactive, it means that that's the wrong one. And here what we see is that the number of inactive responses were also increased, indicating a non-selective behavioral activation. However, because this number of responses remains significantly higher with CNO, it indicates that stimulating the VP elicits drug thinking rather than non-specific motor activation. So considering that the majority of VP neurons, as I was saying, are GABAergic, authors hypothesize that stimulating VP GABA will have the similar effect observed with the global VP stimulation. So to do so, they, in that case, transduce VP GABA neurons with a Cre-dependent GQ dread, so using the VUGAT iris-Cre line, prior to the cocaine self-administration training. So when looking at the responses, a significant increase in nose pokes was observed following CNO injection before the late extension. However, CNO injection did not significantly potentiate drug thinking during the reinstatement. Authors next expressed the GQ dread in the GABAergic subpopulation of VP-PENK, and following extension learning, chemogenetic stimulation of VP-PENK increases responding during both the extension and both the reinstatement, suggesting that VP-PENK neurons constitute a subpopulation of GABA that can facilitate cocaine thinking. They finally investigated the effect of stimulating VP-GLU, so the glutamatergic neurons of the ventral pallidum, and activating glutamatergic neurons significantly reduced active nose poking during extension and cocaine thinking during the cocaine cue thinking. So to summarize this experiment, stimulating VP-GABA did not potentiate drug thinking in the presence of conditioned drug use. VP-PENK neurons constitute a subpopulation of GABA that can facilitate cocaine seeking. And cocaine seeking is reduced by stimulating VP-GLU. So others were then interested in looking at the single-cell calcium imaging and used the Inscopix and VISTA system to visualize large-scale calcium imaging in the ventral pallidum during the self-administration and extension task. So they used the NAV1 HC diogchem6 fast that was injected into the VP using a 0.6 by 7.3 millimeter green lenses. And these green lenses were implanted above the VP. Following 3 to 4 weeks post-implantation, mice were exposed to self-cocaine administration, extension, and cue task, here in panel B. Here, responses and calcium events were collected at 3 different time points during the protocol. So during the first day of extension, that we'll call extension 1, after 10 days of extension training, that we'll call extension 10, and during cue-induced cocaine thinking, so during the reinstatement. So mice showed active nose poke responses and cocaine infusion during this self-administration and during extension protocol when wearing a demiscope, although the camera appeared to reduce responses in extension 1, but the responses during both extension 1 and reinstatement remain elevated, significantly elevated. So when looking at the calcium signals, so calcium events here were identified using the Inscopix mosaic software. Panel D shows a nice representative calcium traces recording from the different VP subpopulation. So when looking at the frequency with the different subpopulation, we see that in VP-GABA, the frequency of calcium events was selectively increased during the reinstatement. The frequency of VP-PENK calcium events did not change across the sessions. And by contrast, VP-GLU neurons were more active than VP-GABA and VP-GLUT in overall already, regardless of the session. And this increase is more pronounced during extension 10 and during the reinstatement, and appear actually right after the extension learning, so right after extension 1. Then author looked at the cumulative events distribution to investigate at what time point changes in calcium events occur within each cell type. Now, first, when looking at the cumulative events distribution, we basically found the same results obtained in panel E. So with this significant increase the reinstatement for VP GABA and this significant increase in the frequency of VP GLU during extension 10 and reinstatement. And when looking at the time point when changes occur, a difference in calcium events rate became apparent here and here around 30 minutes for both the VP GABA and the VP GLU. So taken together, cued thinking activates VP GABA by increasing their calcium events. And VP GLU calcium events occur more frequently following the extension learning. So although the increased occurrence of calcium events in VP GLU neurons is consistent with the inhibitory effect of this population when authors used the GQ DREAD, authors decided to look at the activity across the epoch, so between 10 seconds before and 20 seconds following an active no-spoke in extension 1 and reinstatement. So at 0 here, basically the 0 represents the no-spoke. They then look at changes in calcium activity around no-spokes in the different VP subtypes, VP GABA, VP PANC, and VP GLUT. So to quantify the population response magnitude, average calcium imaging was compared, so 2 seconds before and 2 seconds after the no-spoke for each cells. So they first noticed that the population of GABA, basically the population activity of GABA was poorly organized around no-spoke. However, the VP PANC and VP GLU activity was changed around no-spoke. So you can see that for VP PANC, there is a correlation between the population activity and the no-spoke during reinstatement. And here, a correlation between the population activity of VP GLU during extension 1. And here is the population response, so expressed in a unit of this score in the different subpopulation, where we can see the significant increase during extension 1 for VP GLU and reinstatement for VP PANC. So then by organizing individual cells from the differentiation into IMMAPs, so here are basically each line represents one cell, and they basically plot for all cells registered for each different VP subpopulation. They were able to determine the proportion of cells with increased or decreased responses to no-spoke. How? So they basically they use the calcium signal with this same time window, and then so this calcium signal within the same time window were compared, and significantly modulated cells were defined as decreased if the standard if it was lower than 2 standard deviation, and defined as increased if calcium signal was higher than 2 standard deviation when comparing this signal with the baseline prior to the no-spoke. So what they found here is that during extension 1 and reinstatement, VP GABA and VP GLU population contain equivalent proportion of increased and decreased cells. However, the proportion of activated and inhibited VP PANC decreased significantly during reinstatement compared with extension 1. And so that was actually surprising. And so here authors hypothesize that this reduction in the size of the population of VP PANC neurons after extension may be due to a loss of neurons that responded to thinking in a context-specific manner after that context had been extinguished. So to summarize, could thinking activate VP PANC by changing calcium population activity around nose spokes. And the VP glue population activity is elevated around nose spokes selectively during early extension. So in summary, by using a combination of tracing, in situ hybridization, chemogenetic stimulation using GQ-DREAD, and calcium imaging to monitor spontaneous activity, we can authors conclude that VPGU activity reduces motivation, that this process is recruited during the extension learning, and finally that VP GABA and VP PANC, so the subpopulation of VP neuron GABA and VP PANC activity facilitates the motivation to seek cocaine in response to cocaine-associated cues. Thank you for your attention.
Great, thank you, Jonah. This was a really nice study. I think that utilized a lot of different circuit mapping tools to really dissect the role of GABAergic and glutamatergic neurons in the VP during cocaine relapse behaviors. So I think you did a really nice job of walking us through the figures. So we can go ahead and start with the Q&A. So again, we just want to encourage everyone to submit a question using the chat box at the bottom of the screen. So I guess the first question, what is the main target brain region of these glutamatergic and GABAergic VP neurons? And is it known the role that the target regions play in relapse behaviors?
Yeah, so I know about I think it's the GABAergic projection. I know they project to the VTA for sure, yeah. And basically activation of these neurons have been involved in directly in the reward.
Right.
Then about the enkephalin neurons, I know that there is something linked to the so during cocaine withdrawal, there is a surrounding, let's say, enkephalin tone that basically will so the enkephalin will so you have some receptor of enkephalin on the, I think it's the D2 MSM population, and that will basically induce this disinhibition of the neurons in the VP.
Yeah, I think it could be pretty interesting to use maybe like a retrograde GCAMP approach and actually image the subpopulations of neurons in the VP that project to different yes. areas. I think could be really helpful yes. to sort of keep this apart.
Yes. Yeah.
Okay, so another question coming in, were any other behaviors analyzed in addition to nose pokes? Did manipulation of different VP populations alter behavior generally?
So in that case, no. You mean outside, sorry, can you repeat the question, the beginning?
Yeah, so the first part of the question is, were any other behaviors analyzed in addition to the nose poking behavior?
Mm-hmm. No, not in this study. I guess they did some locomotor activity because that's what people usually do when they run this kind of test, but it was not in the study. But I'm quite sure they did that, knowing the data.
Yeah. Okay. So another question. So this paper shows opposing roles of GABAergic and glutamatergic populations towards cocaine seeking during withdrawal. Do you think this circuit is specific for extinction behaviors, or would it also potentiate cocaine self-administration during normal drug-seeking behaviors prior to withdrawal?
That's a good question. So that's a good question. Well, first of all, they are, and that's actually in their discussion. So a part of the GABAergic neurons are basically these enkephalin neurons, and there are also non-enkephalin neurons. So because here basically when we look at the activity of VP-PANC, we see that there is a slight increase of this frequency during extension 1 and extension 10. So it's not significant, but there is still and so author hypothesized that they might another maybe subpopulation of neurons might be involved in this. So you were talking about reinstatement here, right?
So I think the question specifically talking about, well, they didn't record during just the initial self-administration period, like the 0 to 12 days. So it's at least unknown from this figure how these neurons would respond during that particular part of the task. But yeah, I'm wondering if there's some other literature out there about how these neurons would respond during the self-administration aspect of the task.
No, but no, with you mean with calcium imaging in vivo?
Yeah.
So during exactly the self-administration, I don't know. Well, I would say that we will have an increase in which subpopulation, I don't know, and that's something I think that we should ask to the lab. But for sure, we should have some activity at least during at least in the GABA population, subpopulation.
Yeah. Interesting. So I have a question. So I think it was in figure 1, they showed that 5% of these VP neurons co-express GABA and glutamate. I think this is sort of like a recent, discovery in the field that a lot of these subcortical regions co-express both GABA and glutamate. And yeah, so that's 5%. It's a small amount, but given that the VP is playing sort of opposing roles on cocaine relapse behaviors, I'm wondering what you would hypothesize these neurons that co-express both, how they would, what role do they play in relapse behaviors?
Yeah. So what actually they discuss in the paper is that if you look at the results they obtain with GABA, in some cases is not, especially when you look at the reinstatement, you see that there is no significant increase during the reinstatement, and basically the GABAergic, I would say the population of GABA in the VP is really, I don't remember the term they use, but you have multiple population, and it's difficult to basically say. Maybe you can dissect different subpopulation of GABA again, and probably that's why they have this non-significant difference here. So because heterogeneity, there is this heterogeneity of GABAergic neuron in the VP. So about this co-expression of GABA and glutamate, so the question is, like, do you think if I think that they are involved in a particular, because I would say that based on the opposite role here, at least in the VP, I would say that, or I guess that this population that express both are not involved in at least extension or during the Q. That's what I would say. Then I remember that there was a paper actually from the Kalaevas lab. No, not from the from Kalaevas lab on that. And but it was unrelated to I think to the self-administration behavior.
Yeah, I think it's always a head scratcher, these recently discovered neurons that contain both GABA and glutamate. There's a lot of instances subcortically where there's these divergent behaviors, opposite behaviors from GABA and glutamate neurons. So it's always kind of a question as to what these populations are doing with that have both and why are they there? Okay, so another question, are these circuits and cell types specific to cocaine, or do they participate more generally in addiction type behaviors?
So that's a good question. I would say maybe other. So in that case, I would say other drug of abuse that use the same pathway. So it's not only cocaine. I would say also other other pathway. But yeah, I don't think it's really specific to cocaine. In that case, the test they used is specific to cocaine. But I guess the pathway they described should not be only attributed to cocaine, but other drug of abuse. That's what I would personally say.
Yeah, I would agree with that. It's kind of, the question is why did this circuit evolve to respond this way? I mean, most likely it's for naturalistic, rewarding behaviors, and the cocaine kind of just hijacks this circuit, takes advantage of these, the way it responds. Yeah, I would agree with that.
Then also it depends of the, I guess, the effect of the drug itself. So cocaine is, if I remember, acting on presynaptic receptors, and basically cocaine inhibits the recapture of dopamine. Then maybe for other drug of abuse that basically have a different action on this circuitry and pathway. So maybe they might observe like a different results, but I think that in overall, what in this paper they describe could be like I think attributed to different other drug abuse, but the same pathway.
Yeah. Yeah. Okay, so I think that was our last question. So we'll go ahead and wrap this up here. Thanks again, Jonah, for walking us through this really interesting paper.
Thank you for joining.
Yeah, thanks everyone for attending and sending in some questions. I think we had a nice discussion here at the end. We hope this was scientifically stimulating for everyone, and that you'll join us again next week, where we will be reviewing another great publication at the same time next Friday. Please stay safe and healthy, and hopefully we can all get back to our regular research activities soon. Take care.
Bye-bye, guys.